Hello, everyone. My name is Martin, and I'm currently a postdoc at the Karolinska Institute in Stockholm. And today I'll present to you some of our latest research, which we call the pupillary-respiratory phase effect. And I think it ties in nicely to the previous two speakers. So this study has its background in the long-standing idea that our respiration can influence our pupil size, and more specifically that as we inhale, our pupils dilate, and as we exhale, our pupils constrict. And when we got interested in this topic a couple of years ago, we decided to start out by doing a systematic review of the literature to see what is actually already known about respiratory influences on pupil size dynamics in humans. And to our surprise, we actually found that the evidence is not that conclusive and that many important things have not been investigated at all. For example, differences between nose and mouth breathing. So we decided, okay, let's try to figure this out. But why would respiration affect pupil size in the first place? Well, as has already been mentioned, the locus coeruleus influences pupil size. It can regulate both the sympathetic and the parasympathetic pathway, which regulates pupil size. But the pre-Betzinger complex, which is also known as the respiratory pacemaker, it creates the rhythm for inspiration, also has excitatory projections to this locus coeruleus. So whenever it fires, we not only inhale, but it can also send excitatory signals to the locus coeruleus. Furthermore, for everyone who was here on Wednesday, there are many respiratory brain coupling pathways. Another one I want to point out for this study is the one via the olfactory bulb. So whenever we inhale through our nose, the incoming airflow can mechanically activate the olfactory sensory neurons located on the roof of a nasal cavity, which in turn activate the olfactory bulb. And this rhythmic activation has been shown to propagate throughout many brain regions, and in recent years has been shown to play a role in a variety of functions and behaviors as well. So we started by collecting data from 50 people at rest. We collected both their respiration and the pupil size. And the experimental conditions were quite simple. They were sitting in a chair in front of a computer screen at approximately 60 centimeter distance, where they had to look at a fixation cross. And we asked them to come in twice, once while they were breathing through their nose and once while they're breathing through their mouth. And here's some initial results. The figure is a bit complex, so I'll try to walk you through it. In green, you see the results for nose breathing, and blue for mouth breathing. And basically here we have the respiratory cycle, with at 0 degrees being inhalation, 90 degrees is peak inhalation flow, 180 degrees is exhalation, and 270 degrees is peak exhalation flow. These individual lines on the inside is the normalized pupil size from all the individuals. And as the lines come closer to the inside of the circle, pupil size is smaller. And as they go further to the outside, pupil size is larger. And what we did is we divided the breathing cycle into 18 evenly spaced bins of 20 degrees each. And if the bin is shaded, it means their pupil size is statistically different from what would be expected from a normal or a random distribution. And if the shaded bin is on the inside of the circle, it means pupil size is significantly smaller. And on the outside, it means it's significantly larger. So you can see that for both nose breathing and mouth breathing, for the majority of the participants, pupil size tends to be smaller during inhalation and larger during exhalation. Furthermore, on the outside of the circle plot, you have a polar histogram, and this shows the proportion of participants that have the mean vector direction point towards a certain breathing bin. This does not take into account vector length, but just vector direction. A somewhat simpler plot is here, which is the average normalized pupil size across participants for both nose and mouth breathing. Again, 0 means inhalation onset, and 180 degrees is exhalation onset. And you can see here that quite nicely pupil size varies systematically, with largest pupils usually around mid-exhalation, and smallest pupil size happens around inhalation onset. Finally, we also looked at change in pupil size, so pupil dilation and pupil constriction, by taking the pupil size derivative. Everything above the 0 line means pupil dilation, and everything below the 0 line is pupil constriction. So you can see that pupils dilate and constrict during both in and exhalation, but the vast majority of pupil constriction seems to occur during the end of exhalation, and for most of inhalation, our pupils dilate. So we thought, okay, this looks nice. Let's see, can we replicate this to make sure it's not just a spurious finding? So we collected an additional 50 participants. We pre-registered our analysis methods from the first part, and we were happy with showing that the effect replicates very well. So then we thought, okay, let's take it one step further. We can show this pattern at rest with what happens when people are actually engaged in a task, and specifically a visual task where pupil size actually matters. Is this more similar to the hippus, which tends to disappear as people's arousal increases, or is it something that is more stable than that? So we collected 112 participants. The people from experiment 1 and 2 were participating in this as well. The task conditions were basically the same, but now we have a 45-minute recording time, and people had to do a visual detection task, so they had to detect faint Gabor patches. And not only did the effect not disappear, it became way clearer, and there's now less variation across participants as well, as you can see in these graphs. And we attribute this mainly to the longer recording time. So we think that this effect that we see, it's relatively, maybe it's not very strong. There are a lot of things that affect the pupil size, but over time, this pattern clearly emerges. To continue, we decided to check, okay, is there a difference between passive breathing and active breathing as now other brain regions start to become engaged? So this is actually a sneak peek at a data collection that's still ongoing, but 33 subjects have been analyzed so far. And we asked participants to either breathe freely or breathe at a slow pace of 8 breaths per minute or a faster pace of 16 breaths per minute, guided by an auditory cue. In addition, we changed a few other conditions. Now the room was brightly lit. We know that background light affects the pupil size very strongly, so does that have an effect? And we also increased the fixation distance. We used a mirror setup and asked them to fixate at a cross at approximately 3.5 meters distance, as also the fixation distance is known to affect our pupil size. And if you fixate at a more distant point, then your eyes relax a bit more as well. And here again, we find the same pattern basically as before across conditions. The change in pupil size looks a little bit different, but is mainly driven by this peak here during slow breathing. Otherwise, the change looks quite similar to what we found in the previous setups. And also the effect size seems to be a bit larger now compared to the previous studies. And we guess this is because the eyes are more relaxed as they fixate at a more distant target. In addition, we have a more updated eye tracker in this setup, and we now can get accurate readouts of absolute pupil size values. So here we also have an idea of the effect size. And in this, for these participants, it seems to be that the pupil size fluctuates around 0.2 millimeters, which is around 4 to 5% of the pupil size. So it's not huge, but it's not nothing. Finally, because we are an olfactory lab, I went back to some data that some colleagues of mine collected a couple of years ago, where they were studying people with congenital anosmia, or people who were born without a sense of smell. And in particular, these people don't have a sense of smell because they're born without an olfactory bulb. So basically, we have sort of a lesion model to see whether the olfactory bulb plays a role in this effect. And here I managed to analyze 34 people, 13 people with a normal sense of smell and 21 without olfactory bulbs. Otherwise, the conditions was again different lighting, different fixation distance, and now they were lying down because it was in an MRI scanner, and 45 minutes recording time approximately. And lucky for me, they collected eye tracking data and breathing as well in that study. And also here, we find the same effect. It's very clear. So olfactory bulb is not necessary for this. And besides like an olfactory bulb, these people are completely healthy. So to conclude, what we call the pupillary-respiratory phase effect, maybe come up with a better name, don't know. But over time, pupil size seems to fluctuate in a manner that's predictable by the breathing cycle, with largest pupils during exhalation and smallest pupils around inhalation onset. Furthermore, most of inhalation consists of pupil dilation, and the majority of pupil constriction occurs during the second half of exhalation. And this effect seems quite robust as we tested it across a wide range of conditions. However, there are some limitations. It is definitely not found in every breathing cycle, and we also don't find it in every participant, though in the majority of participants. And the main outstanding questions for me are, what is driving this effect, and is the effect strong enough to influence our visual perception and our behavior? And this is something we're currently investigating. With that, I want to thank my collaborators on this project and all of you for your attention.
Great talk.
Thank you for a great talk. Well, yeah, actually I have several questions. But so firstly, I'm interested in the individual difference, especially like aging would affect this breathing pupil in interaction.
So your question is if I'm interested in individual differences between
Yes, individual differences.
between participants, you mean?
Yeah, I mean, so for example, I think that the older adults are has likely to the smaller pupil size, and so you mean not every participant show this effect, and so what kind of participants are show greater effect of this phenomenon?
Yes, that's a very good question. So why do some people not show this effect? I'm definitely interested in that. I've done some quick looks into it, and I'm not sure so far. I'll look a bit more at the breathing characteristics. But so far, my guess would be that that probably it shows in all people if you record long enough, and that just some people are maybe mentally distracted or something, that just other things that affect pupil size more are interfering with this effect. But I don't know yet. Is the short answer.
Yeah, it's interesting because it may be related to the mechanism that the respiration affects pupil. Okay, thank you.
Other questions? Yeah.
Yeah, thanks for the talk. Can you speak to the effect size that you showed? Because it looks incredibly consistent across the experiments that you just presented. So the effect of pupil size over respiration phase across the bins. But on the y-axis, the z-scores were really, really low. And I don't know what you were plotting, whether that was SEM or something around so what kind of variation measure you used? And you didn't speak to the statistics of how you actually assessed whether there was meaningful cross-participant variation across the respiration cycle?
Yes, so good question. So the shaded lines is confidence intervals, 95%. And for the shaded bins to see which were statistically significantly different, we used permutation testing. So we took basically the data from all participants. We looked at how many data points are in a breathing bin, one of the 18 different bins. And then if you take pupil size randomly from all participants, independent of where in the breathing cycle the pupil size is measured, take an equal amount of numbers of data points, and we compared it to that, if that makes sense.
In the sense that I get what you're saying, yes.
Okay.
I think.
But otherwise, it doesn't make sense.
Yeah, okay. I got to think about this. Give the cube to someone else.
I'm arriving with the cube. No worries. Thank you. There it is.
It's an incredibly naive one, but how do you measure the pupil dilation? What kind of eye tracker do you have? I'll tell you why.
For these experiments, we have a gaze point eye tracker. For the one in the scanner, we have a different one. I don't remember the name right now.
So this is the one that is, sorry, you've brought it for no reason. This is the one that is far away from the participant. It's not attached to the eyes.
It's not attached to the eyes, but I wouldn't say it's far away.
I'm only wondering because indeed, when you breathe in and out, there is movement. So if you're tracking the pupil based on an ellipsoid or even a centroid, the movement itself will actually modulate, no? Whatever the measurement of the pupil is. I don't know, I'm naive on this, but it's just a...
You mean the movement
the
of the
physical movement that occurs as I breathe in and out, no? Maybe I'm getting this wrong. This will move. No?
I'm not sure.
I don't know anything about pupils.
My naive intuition would be that any kind of movement, as minor it is, would be vertical and should not affect the estimation of the pupil size per se. So it could be a bias in slight eye movements, but also would not have that rapid trajectory that usual eye movements have. And usually estimation of the pupil, given these fluctuations, should be reliable enough. Depends on the system, depends on many things, but...
Yeah. And since we find it in different, we find it both seated and lying down, so there's also, I don't think it should affect it, but good question.
Legit.
Yeah.
Thanks a lot. Wonderful talk. I was wondering when you do the paced breathing and whether you could speculate whether you would expect changes in effects if you, for example, if participants, for example, repeatedly do different breathing patterns, whether you would expect that there is a change somehow in the pattern you see, or whether you would rather think that this stays really constant.
So if they would keep switching between paces over a longer period of time, or?
If they would basically train a certain breathing pattern, because I guess if you do paced breathing at the beginning, it might be, and it is not your natural rhythm, right? It might maybe be different than when you, for example, practice a certain breathing rhythm, I don't know, in meditation or whatever situation, yeah.
Yeah, so I can explain a bit more. So here, first we're breathing freely, and I think in the subjects I look at, the free breathing frequency on average was like around 11 breaths per minute, so quite well in between the slow and the fast breathing. And the slow and the fast breathing were right after each other. But no, I don't think that, I wouldn't know why it would change if they would do this over a longer period of time.